recente, com uma grande expertise, né, toda essa parte da pesquisa, desde o microbioma, né, a análises epidemiológicas. So now, Lorena, you can start your presentation. She'll be talking English, but feel free, as I said before, to make questions in Spanish or in English or even in Portuguese.
Hi, everyone. Hi, Juliana. Thank you so much for the introduction, and I hope everything is set up. You can see my screen. You can hear me well.
Yeah.
Okay. And as Juliana said, I'm Mexican, so I speak Spanish. I understand Portuguese. I don't speak Portuguese fluently, but if there's any questions, just feel free to put them in the chat. I'm not checking the chat right now, but feel free to interrupt if there's something pressing that might be coming. I'm very happy to be here. I'm very happy that Juliana invited me to share with you my research today as an example of applying what you've learned so far about this awesome and mysterious world that is the microbiome. I'll share a little bit. My presentation is not very long. I'll share a little bit of the introduction and then share what I've done so far in my PhD. As you might have seen throughout this course, we have a lot of details on how the microbiome changes in early life, and I'm sure you've discussed this plenty before, so that the biggest microbiome exposure happens at birth, and that it continues through infancy and childhood. So it's affected by multiple factors. Among them are maternal infections, pregnancy length, the type of delivery, the place of delivery of the child, also the use of antibiotics, and later on some feeding approaches, so breastfeeding, formula fed, the time of introduction of solids, and as well some more broad terms like lifestyle, geography, and socioeconomic status. So these early years of life are a great opportunity for modulation because they can determine the microbes that will colonize our gut and stay with us throughout adulthood. And this is why this period is considered critical for microbiota development. And a number of bacterial signatures have been associated with either undernutrition or healthy growth in these first 1,000 years of life, which is like a topic on itself, research on this area. So in parallel with this stage of life is also a critical period for growth and development. So physical growth, we defined it as the increase in size or mass of a child. So this process is dynamic, it's very complex, it's very long, and it goes from infancy through childhood and adolescence. So we know now that growth is globally recognized as the best indicator for physical well-being in children, and it can also help us measure disparities that are faced by populations. So we routinely track childhood growth as an indicator for optimal health and development, and this is often reflecting of the nutritional status of the child. So tracking can help us predict health outcomes later in life, such as a rapid weight can increase the risk for non-communicable diseases and heart disease, such as diabetes and heart disease. And then impaired growth can show to affect cognitive development long-term and increase morbidity and mortality. So as I mentioned, growth is a process. It's continuous, but it's also non-linear. So this means that it doesn't just follow a perfect line. So what we can use are growth curves or growth trajectories that can assess individual growth, but we can also use growth curves to assess the growth within a population. So here I'm presenting the WHO growth curves from birth to 5 years of age for both females and males. And these are used as a reference to assess individual childhood growth. So here, the green line, which is in the middle, represents the normal growth based on a reference population of healthy term children. So we can see that the first 2 years of life are characterized by a very intense, rapid growth, both in height and weight. So here I'm just summarizing for the BMI. And a lot of research, this is why a lot of research has focused on this stage, because this is when the fastest growth that we will have in our postnatal lives happen. However, what happens after 2 years? So we see that it kind of slows down. It doesn't stop. We don't stop growing right away, but it slows down, and there's still multiple growth spurts after it, but they're shorter. And but the last and the second most important growth spurt happens around 9 to 15 years of age. So these are the growth curves for after 5 years, so 5 to 19 years of age. And we see that there's this other peak in growth that happens around 9 to 15 years of age. So it happens a little bit earlier in females and males. And then we can see that growth kind of like stabilizes through adulthood. So this growth is more steep in females and males, and this is the stage that is considered adolescence. So the WHO defines adolescence as the period between 10 to 19 years of age. And adolescence has been historically under-researched, and this is a concern that was brought up by the research community previous years. So a few years ago, there was this call to increase the attention to the importance of research in adolescence from major journals. And since then, the research in this stage of life has kind of like increased after that. So this is especially important because adolescents represent 16% of our population, the worldwide population. And in countries such as Brazil, which is the fifth most populated country in the world, adolescents represent 14% of this population. So we definitely need more information on this stage of life. So I mentioned the WHO defines adolescence at the stage between 10 to 19 years of age. And this is a critical period for exponential physical growth, like I mentioned, in development. And then when a child's sexual and physical characteristics mature, that's when they reach puberty. So there's no specific age for this. However, it happens in all adolescents, but at different age, duration, and time. So it's very variable within a population. So puberty is characterized by the rapid maturation of metabolic, endocrine, nervous, and immature immune pathways. So these strongly support the growth and development of the child. So puberty involves sexual maturation, involves an increase in height and weight, a completion of the skeletal growth, and also changes in body composition. So you can see that all of these will increase the demands of nutrients in a child. So like I mentioned, we have the velocity of growth is very high early life. And then we have the second growth spurt, which is the last one in adolescence. So this happens at the onset of puberty. It begins about 18 months earlier in females than in males. And is part of this large collection of physical manifestations of sexual maturation. So in females, includes breast and genital development, and pubic and axillary hair, and menarche as well, which is when girls get their period. And in males, this includes genital development, pubic, axillary, and facial hair. So the changes that happen in puberty, they can be assessed by Tanner stages, which classify puberty in stages from 1 to 5. So 1 being pre-pubertal stage and 5 being adult stage. So these are currently the most frequently used classification to assess stages of puberty in epidemiological studies. So we know that pubertal growth is strongly regulated by genetics, which can explain from 50 to 80% of the variation in the timing of puberty, but also nutritional status and environmental factors can predict when puberty happens. So one environmental factor that may have a role in dysregulation is the microbiome. So it can, because it has this role in processing nutrients and providing energy to the host, so it can be also influencing growth as well as the production of hormones or the metabolism of hormones. So many important and profound changes in physiology occur during puberty, and this also may be the case with the gut microbiome. So we can see that growth and microbiome development, they are kind of running parallel, and they are highly intercorrelated. So if there's disturbance in either one of these, it can affect the trajectory of growth of the other and overall the development of a child. So we know that in adulthood, the microbiome is kind of relatively stable. We have observed that there are still factors that can affect the gut microbiome composition throughout life. And some of these factors include the use of antibiotics, the diet, genetics of the host, the environment, their socioeconomic status, also ethnicity, physical activity, and of course, state of disease. And it was previously believed that the microbiome was established by age 2. Then some people say age 3. Now there are people saying like established by age 5. So, but basically they're reaching conclusions based on when the diet is kind of established or it's more similar to that of the adults. However, in the last few years, there's been research that has compared the microbiome of children versus adults and has find some differences. So usually it was previously believed like, oh, after 3 years, the microbiome just reaches the stable state and it's similar to adults because they were not including a lot of children or adolescents. So they were kind of pulling together adolescents with adults or adolescents with children. So observation, like they were not able to observe differences between these populations specifically. So research has just now started peeking through the microbiome in adolescents. And subsequent studies have consistently find that there are differences between adults and children. So there's also this lack of studies that have analyzed the effects of hormonal changes in puberty. I understand this is very difficult to do because all that it entails, and as I said, the timing of puberty is different in every child. So, but being difficult doesn't mean it shouldn't be done. So we still need information on this side. Also, the studies that have done it, some of them in animals have shown that the microbiomes are essential kind of intermediaries in energy harvest, in fat storage, in regulation of inflammation, the gut barrier function, but also interactions with the host genetics. So some of the first evidence that the gut microbiota maturation was related to childhood growth came from a study that was done in a group of twins from Malawi. So these were undernourished kids, and the gut microbiota was assessed for a little over a year, so from 0 to 32 months. And they found that there was this microbial signature associated with age. So you can find distinct bacteria that will change with as the children age. And then they did this kind of preclinical assessment. So they transferred the microbiota of children that were underweight and children, that were healthy. So they were kind of pairs of twins. They transferred it to mice, and then they saw that the microbiota of healthy children, they like help mice develop nicely. However, when they transferred the microbiota of undernourished children, they observed that the mature microbiota of the donors was also reflecting an impaired growth in the mice. So there was weight loss, and there was disturbance in carbohydrate and amino acid metabolism in the mice as well. Then when they housed them together, the healthy and unhealthy microbiota mice, they saw that the microbiota of the healthy mice was transferring to the unhealthy ones, and it was kind of avoiding these, preventing these growth impairments. So the mice were developed nicely after that, even though they initially had the unhealthy microbiota. So from this study, they concluded that the microbiota was causing malnutrition. And then research has been followed after that. So then they kind of derive like a C-score of microbiota for age C-score, kind of like trying to use the similar to BMI and height for age and all these. Based on the top tags that they observed that were changing. However, this hasn't been fully translatable in other studies yet, so it's still kind of like up to debate. But they did try to do that. So This just shows like how dynamic and how diverse and different microbiomes are throughout populations and different conditions. So another study that also highlights the differences between nutrition and the gut microbiota was one from Mexico, where they assessed the difference in composition of the gut microbiota in children classified as both undernourished and with obesity compared to this normal BMI group. So the normal BMI group consistently had higher richness and higher diversity compared to the two groups. And we can see that specifically the obese group had lower diversity. However, the undernourished group had lower richness. So that's why it's important to observe multiple metrics of the gut microbiome and not just focus on one, because they can give you different information. Moreover, they also observed that children with higher abundance of firmicutes, so undernourished children had higher abundance of Lactobacillus, and Proteobacteria was represented in the obese, underrepresented in the obese subject. So it can be concluded that the course of the intestinal microbiota succession plays an important role in the regulation of early life growth. Moreover, we look at the research that has been done in differences by sex. So the first study that highlighted these differences was a study in mice. They assessed mice at different developmental time points, and they found that sex-specific differences became more evident in puberty, and then most apparent in adult mice. So puberty for mice was around 6 weeks of age, and then 14 weeks is already adult mice. So you can see that they kind of start diverging at 6 weeks by sex, and then at 14 weeks they're more separated. So this plot show that relative abundance of these mice. And this suggests that different stages of puberty might be associated with different microbiomes. And regarding diet, there's evidence that the microbiome is different according to the geographical setting and the diet of adolescents. So there was a study that was carried out in the Philippines, and they assessed the intake with an FFQ of children 7 to 9 years old. They were from 2 different areas of the island and identified different microbiota patterns. So one, they were associated with the macronutrient intake. So they were clustering separately those with a high carbohydrate intake than those with a high fat and protein intake. So the correlation analysis showed that the nutrient intake explained 13% of the variation in the microbiota, which taking into account that variation in the microbiota depends on multiple factors, I think kind of high. So most of the variation was explained by the city of residence. So in these studies, factors such as socioeconomic status, pet ownership, living conditions, and age have been found to have a significant effect in the microbiota composition, as well as the nutritional status. So we know that the study of trajectories allows us to highlight these early life risk factors that can predict and play a role in diseases later in life. And it has been demonstrated that the gut microbiome in early life can predict later growth. So if healthy dietary habits also are established during childhood and adolescence, they can continue into adulthood. So we know these 3 factors are dynamically linked. And this is especially interesting in puberty, which is the stage with exponential physical growth and sexual maturation. However, there's still under investigated this topic. So this was the objective of my thesis, to look at the association between the growth trajectories, the gut microbiota, and diet. And we did so, as Julian said, by leveraging the Pelotas-Berg cohort. So maybe you're already familiar with the cohort. So since 1982, 4 cohorts have been carried out in the city of Pelotas. And they have been recruiting all the birds that happen in the city in the specified year and then following them through life. So we use data from the 2004 cohort, and the objective of these cohorts specifically was to investigate the impact of early life exposures on health outcomes and to study inequities on health conditions. So this cohort has collected perinatal information and then follow them the information that we use: 3 months, 1 year, 2, 4, 6, and 11 years old. So including maternal information, socioeconomic status, children's anthropometrics, dietary intake. So we use first the anthropometric measures from these the previous follow-ups up to 11 years old to model the growth trajectory. So this was the first objective of my thesis. And we published the results of this objective in scientific reports. So we used a shape-based approach to cluster growth trajectories from birth to 11 years old, using both BMI for HC scores and the height for HC scores. So the approach that we used included also an age correction to account for gestational age. So as I mentioned before, the WHO growth standards are based on a reference population of term-born children, so not including preterm in their overall reference. So it has been highlighted before that these standards might not be suitable for application in children that are born preterm. So we used an age correction, and this age correction can better estimate the C-score values of these children. And this is usually done routinely in clinical settings, but in epidemiological research is not as often seen. So this age correction consists of calculating the difference of gestational age at birth, then subtracting this number from the postnatal child age. So in this example, I put here a child that was 8 weeks old and born at 35 weeks of gestational age will be compared to a 3-week-old children instead of an 8-week-old children. And this will drastically reduce the number of children classified with low C-scores in a population. So we did this for all the preterm born children, and along with the shape respecting clustering that we used, we identified 3 growth patterns in each. So in the BMI and the height for HC scores, and we labeled them as increasing, stable, decreasing, or high, middle, and low. So the middle pattern was just a stable pattern. Then we observed this increasing or high velocity trajectory, and then this low or decreasing velocity. So we observe here that most of the change, as we know, happens in the first 2 years of life, but then we were able to capture the variation that happened within before the trajectory kind of like establishes throughout. So we use these clusters to then assess the microbiome, but the utility of using this kind of approach was that we were able to identify a group of children that were at high risk of remaining undernourished later in life. So in these graphs, I show the comparisons that we did in the study. You can check it. We took the total. So here I'm presenting the total percentage of children classified as stunted, and we see that the overall proportion when we used gestational age correction. So these are gestational age corrected. So in all children, when we use gestational age correction, the percentage of preterm born children is drastically reduced, specifically in early life, because that's when weeks matter the most. But also, this effect decreases with age. So that was the first result that we found. And then if we look at the pattern membership, so where these stunted children are, we see that the approach that doesn't take into account the shape of the trajectory put children in the low trajectory. However, when we do take into account the shape of the trajectory, children kind of get redistributed throughout the 3 trajectories. So we kind of so the trajectories are more taking into account the overall growth of children and not just based on the one-time classification of stunting. So we wanted to see if this was mostly because of the children that are born preterm, and interestingly, it was not. So we found that we did not only reduce the proportion of children classified as stunted in early life, but these also carried throughout childhood in the preterm born population. So overall, we detected this group of children that were high risk of remaining stunted later in life. So in this case, will be children that were in the low pattern, so they were consistently undernourished. So moving on to the microbiome part. So what I know that was not related to microbiome, but that was part of my study. And that's carry out to my second objective of my thesis. So here we analyzed 331 samples of children from the Pilotusburg cohort. And this was done at 13 years old. So we pre-selected as a sample of 500 children based on their anthropometrics at 11 years old. So we wanted to overrepresent all the groups of BMI classification and hyperage classification. So to be able to account for differences within these variables. So we collected stool samples. We also obtained lifestyle and puberty information. So type of diet, using antibiotics, pet ownership as well. But also we did assess their pubertal stage. So as I said, in using the Tanner stages, which go from 1 to 5, and then in females specifically, there was also an option to choose that they were already went through menarche or their period. So we also did obtain information on diet with a food semi-quantitative food frequency questionnaire, and we collected their one stool sample. So then we analyzed the fecal microbiota with Illumina MySeq targeting the B3, B4 region. And then we pre-process the data using the UNOIS pipeline, and we obtain alpha and beta diversity using QIIME2, and then also abundance, so relative abundance information to further assess their microbiome. So we use the growth trajectory patterns that we derived previously to observe the results here. So these kind of like main characteristics by all this the sample, but also divided by the growth patterns, the BMI and the hyperage patterns. And age throughout was similar. There was a similar proportion of females and males. And also, interestingly, so the pubertal stage. So most females had already gone through menarche at 13 years of age. So we didn't get a lot of variation in early stages. So we grouped stage 1 and 2, then stage 3, and stage 4 and 5. And with males, we did see a lot more variation in their stages of puberty. However, they mostly were between stages 3, 4, and 5. So I guess further studies an early age will be best to capture a lot of more variability in puberty if that was the objective to look at in this population. So when we looked at the microbial composition, we observed a similar overall composition of the major phyla and genera among males and females. So there was this gradient inverse relationship of relative abundance, and mostly the major phyla were firmicutes and bacteroidetes. There was none of the factors that we assessed could explain these gradient relationships, so it's just normal variation that can happen in a population. And for the genus level, the most common one were Prevotella, Ruminococcaceae family, Bacteroides, and Faecalibacterium. So this goes in line with the literature that these are the most common bacteria that you can find in human microbiome. So we looked then at the association of the microbiome with sex, the growth patterns, and puberty. So first, we didn't identify any differences among sex. We did observe an overall association of the microbiota with the BMI patterns. So the increasing pattern had lower richness and evenness compared to the middle pattern, which is kind of like the normal one. And only when we did the subgroup analysis, only males showed the same association. So this was not observed in females, and this was only observed for richness, not evenness. So then for the hyperage pattern, we didn't find any differences, and also among pubertal stages, we were not able to observe differences with the population that we assessed. So we then looked at the differences between these groups. So remember, alpha diversity looks at difference within a group, with the variation within a group, and then beta diversity looks at the association between the groups. So we again observed an increase, that the increasing BMI pattern had clusters separately from the other two in the overall analysis, but not in the subgroup analysis. And then we did observe that females in the increasing and decreasing height for each pattern had a different microbiota. And although the microbiota was quite homogeneous with puberty stages, again, we observed that males in a later stage of puberty cluster separately from those that were in the middle stage. So these results were observed only when we used distance metric that does not account for abundance. So this could mean that the observed results are due to genera or taxa that are in a small amount in the gut microbiome of these children. So to compare, there was a study in Canadian children, in Canadian children 0 to 5 years of age, so a little bit younger. And they assessed also the BMI growth trajectories in association with the gut microbiota. So they did so at 3 and 12 months of age. So they observed 4 growth trajectories. So there was like low, stable, like kind of a normative, high, stable, and then they observed like a rapid growth one. And then they assessed the diversity as well. So at 2 time points, at 3 months and 1 year. So overall, they observed that there was an increase in the diversity in the first year. But interestingly, when you look at specifically within each trajectory, the rapid growth pattern didn't change as much as the other two. So the diversity didn't increase as much. And this is similar to what we found. So this group had lower diversity in their gut microbiome. And then specifically when they look at genera, they found that Prevotella, Akkermansia, and Sutrella were decreasing in the first year. And then these 4 other were increasing at the first year. So Streptococcus, Rhamnococcus, Enterobacteria, and Clostridium. So overall, lower change. So they specifically looked then at Akkermansia, and they observed also that these didn't change as much as the other trajectories. So Akkermansia has been previously associated with weight changes and is currently being investigated as a therapeutic for probably a minor weight loss, but still under debate. So just to compare our results. We then look at the dietary patterns of these children. So with the FFQ, we derived dietary patterns. We identified 3 dietary patterns. We named them as processed, coffee, tea, and vegetables, and then a common Brazilian one. So the processed one had fast food snacks, candies, milk, processed meats. And then the coffee, tea, and vegetables was just basically that, coffee, tea, the sugar that you add to beverages and vegetables. And the common Brazilian one was characteristic of beans, rice, bread, and fats. So we identified these patterns and evaluated the participants' adherence to each of them in association with the microbiota. So we observed that the overall richness in the low adherence, so differences only in the coffee, tea, and vegetable pattern. So there was overall lower richness in the low adherence pattern compared to the higher one. And those with low adherence to also the common Brazilian pattern had lower richness and also lower evenness compared to the high adherence pattern. So there was, in the subgroup analysis, no differences in the other patterns, but in the common Brazilian one, the differences were driven mostly by females. So again, lower adherence to this pattern had lower richness and evenness. And then when we look at the differences between the groups, Again, no differences in the process pattern, but with the coffee, tea, and vegetables, we see that children with high adherence to this pattern had different microbiota from those that were in the low and the middle, in the overall, and also in the subgroup analysis. And we also observed that differences among children with the high and low adherence to the common Brazilian one in both sexes were different. And also here using both distance metrics, so one that takes into account abundance and one that doesn't. So we then wanted to look, explore how our variables of interest could be interacting with one another. So we created a path modeling and tested it using PLS path modeling, which is partially squares path modeling. These model include the dietary patterns, the growth patterns, the pivotal stage, and the microbiome composition. So all the models that we tested, so here we have the BMI models and the HyperH models, and all the models that we tested show an association of diet and growth with the microbiota. So diet affecting microbiota and growth affecting diet microbiota. So the continuous lines are significant associations. The dotted lines are non-significant. And these in turn show association with the pubertal stage. So there was, the microbiome was affecting the pubertal stage. So indirectly, the dietary patterns and the BMI patterns were affecting the pubertal stage throughout the microbiota. So this path modeling is an exploratory analysis, is not, it kind of tells us where to look at next. So the one interesting thing that we found is that the OTUs that were associated microbiome with puberty were negative, had a negative load in females and a positive load in males. And this was true for both growth patterns. So there's still, we're still evaluating the results of what this means, basically, because there's more than 100 OTUs that could be explored here. So in each that we found that were significantly associated. So all these results of the second objective are published as a preprint. So if you want to check them out, you can look them up. But so we're just trying to finalize this part to be able to send it for a journal. So overall, we found that the growth trajectory patterns from birth to late childhood are associated with the gut microbiota at age 13. And also we identified these associations among childhood growth, diet, and the pubertal gut microbiota in a sex-dependent manner. And this will be the kind of first study to associate childhood growth trajectories in late childhood and the pubertal microbiota. And also these growth patterns through childhood may help us shape the microbiota diversity in early adolescence. And these might be related or not to the pubertal stage, where we're not able to observe differences in our study. And also, it's important because if we identify these microbial communities that are involved in childhood growth, we may highlight these opportunities for modulation strategies later on. So overall, that's my research. I would like to thank my supervisors, my committee, everyone that has been involved in this project, and also my financial sources. So I'm happy to take questions. I hope I didn't speak too fast. I tend to.
Bom, em primeiro lugar, first of all, thank you, Lorena, for your presentation. I believe that was quite interesting for us to see all this connection between the microbiome data and epidemiological data, and how complex it is to conduct this kind of analysis. As you can see, she didn't use like, she used a very complex statistical approach for this analysis. But, well, we didn't see, let's see if there is any question in the chat. No, exactly. But I will start with one question. As we see in your presentation, the Brazilian pattern, we observe a negative, those with a lower adherence to this pattern, you observe a lower diversity. So you observe effect when adolescents did have a lower adherence for this pattern. So we can we can think about it like our traditional pattern, that have beans. In your pattern, it was rice, beans was more the common one, but we see the effect of beans, for example, that is composing this pattern. But we didn't see any association with the processed one that we could, in our hypothesis, was that the processed one could also affect the diversity. So could you talk a little bit more about your data on dietary patterns and the microbiome? If you want to maybe go through today's slides to show again for the students.
Yeah, that's a great point. And also, so one thing, it was overall very challenging to assess this dietary data, and you're a witness to that. So we overall, in the samples that we collected, so the 300 children within the 3,000 children that participate in the Pelotas cohort, interestingly, we find that their diet was rather very similar. So there were not a lot of variations. So within the main components. So I think it's interesting that the processed food pattern is the first that, like, the one that explained most of the variability, and then we didn't see any differences in the microbiome. And I honestly would not expect that. In like overall research tells us the opposite, right? Like when you do insert these foods in your diet, that's when the microbiome kind of gets disturbed. So it's just like one more, I guess, like adding to the research community, but there's not really, I don't have a good explanation for it. I have I can just like the description that the whole diet was similar within the children. And also we have to take into account that maybe diet is acting not by itself. Like maybe there are these other components that could be interacting with it. So this is why microbiome research is such a complex thing to work on, because you cannot isolate it just like a relationship and say, like, I found this relationship in this research, so then I can just go and apply this into this other population as it is, because you have to take into account all the other factors that interact with it. So I think microbiome is still very early on, but one consistent result is that when you add vegetables to your diet, that improves your microbiome. So that's as far as a conclusion that we have reached at this point. I hope that kind of helps answer the question.
Okay. I would just make some comments for the other students that are not a little bit familiar with our data with dietary patterns in Pelotas, but I will talk in Portuguese, okay, Lorena? Just to make it easier. Então aqui, pessoal, aqui em Pelotas a gente tem as coortes, né, e tem as coortes desde 82, né, então desde as coortes de 82, que agora eles estão com quase, um pouquinho mais de 40 e poucos anos, a gente vem trabalhando com essa questão da análise dos padrões, né, então a gente tem a análise dos padrões do pessoal ali em torno de 20 anos com a corte de 82, depois fizemos com 93, depois começamos a trabalhar com 2004. E essa questão dos padrões, o que a gente começou a observar? Os primeiros padrões que rodaram com a corte de 82, ou seja, os nascidos na década de 80, né, no início da década de 80, os padrões, o primeiro padrão que mais explicava os hábitos era o padrão brasileiro, né? E à medida que a gente foi analisando os padrões de quem nasceu na década de 90 e depois, a partir de 2000, que foi então com a 2004, a gente vê uma mudança do padrão que mais explica o hábito da nossa população aqui. Então, a 82, o que mais explicava é o padrão brasileiro, e a gente começa a ver uma mudança. Então, quando a Lorena então, analisa esses dados, O padrão brasileiro, ele aparece, mas ele não é o que mais explica, né? A gente vê esse movimento, né? Então, essa questão da nossa transição epidemiológica, né, alimentar ocorrendo aqui na cidade de Pelotas. Então, toda essa questão, por exemplo, como eu perguntei aqui para ela, a gente tem muito clareza, né, do efeito, da provável efeito dessa alimentação mais processada, né, pode ter na microbiota, e claro, nesta estudo aqui em específico, com um número menor, né? A gente não estudou toda a coorte, a gente fez essa análise numa subamostra, né? Então, acabou, né, sendo um pouquinho menos de 400 crianças, né? Adolescentes. Então, realmente, foi um padrão, os padrões em si eram os que nós esperávamos, né? Nós já vínhamos vendo essa tendência, então, um padrão processado explicando mais os hábitos, aí um padrão ali um pouquinho mais restrito, né? Café, chá e vegetais, né? Mas então, vegetais, a gente sabendo, então, tem um grupo de alimentos ali, né? Que alimenta um pouquinho mais a nossa microbiota e o padrão brasileiro. Então, em termos de padrões, eram esses padrões que a gente esperava, né? Só que, claro, como a análise dela na parte da microbiota veio desse subestudo com, então, uma amostragem menor, né? E aí, quando a gente pega essa amostra, divide meninos, né? Meninos e meninas, né? E tem essa questão também do estágio puberal dessa subamostra, acaba, né, claro, as análises ficam mais, a gente não acaba não tendo um N tão grande, né, mais provável de observar um dado significativo, né. Então, ao longo do processo dessa análise que a Lorena realizou, a gente conversou muito, né, então, acaba, né, por exemplo, às vezes quando a gente tem a nossa hipótese, a gente tem muito dado já de estudo, né, de modelo experimental, e quando a gente vai estudar o modelo, né, os humanos, né, aí tem tantos fatores, né, como ela mesma comentou, tantos fatores que alguns a gente consegue controlar, outros a gente não consegue controlar, né, como a própria exposição ao uso de antibióticos, né, então a coorte tem muito dado, né, por exemplo, toda a parte de amamentação, né, como foi a introdução alimentar, né, coleta essas informações ao longo do tempo, né, mas a gente, claro, sempre vão ter outros fatores que a gente não consegue captar na nossa análise, né, para ajustar, enfim, para considerar nessas análises, né. Então, quando ela relacionou aqui, então, a microbiota, né, as questões da microbiota com os padrões, né, a gente, né, às vezes esperava um pouquinho mais, né, mas acaba caindo em algumas limitações, né. Bom, então só para dar mais ou menos um contexto para vocês, né? Não sei se vocês também querem fazer algum comentário, outra questão interessante. Então, a Lorena utilizou esses dados, né, desse subestudo, rodando a questão dos padrões e grande foco também na trajetória de crescimento, né, relacionado com a microbiota. Agora, a gente tem uma outra aluna utilizando também esses dados, né, e ela está relacionando a questão da alimentação, né, da dieta, com a microbiota, né? Então, mais focando em dieta e microbiota, mas ela já está fazendo uma análise mais específica dos alimentos ultraprocessados. Então, utilizando essa análise, né, proposta pela Nova, então fazendo uma análise um pouco mais específica ainda também nessa mesma subamostra, né? Mas focando um pouquinho mais para direcionar essa visão, assim, da questão do ultraprocessado para se aprofundar um pouco mais. É até uma aluna que está aqui participando da disciplina também, né? Mas quem sabe futuramente depois ela vem nos próximos anos, né, apresentar os dados dela. Tá. Não sei se vocês também querem fazer outras perguntas, por exemplo, sobre a questão de como foi esse processo de coleta, de desenvolvimento desse trabalho, né, dentro das coortes, né, se quiserem fazer outras perguntas metodológicas, assim, porque também foi um desafio, né, para a gente conduzir esse trabalho de coleta de amostras. Outra questão que foi um desafio na realização desse estudo é que as amostras foram coletadas aqui e foram depois todas enviadas para Toronto. Então, eles é que realizaram todas essas análises, né? Então, teve todo um processo bem burocrático de enviar as amostras, demorou um tempo, né? Para realmente ela ter acesso às amostras e realmente conduzir as amostras lá, as análises, né, lá. Então, é um processo, né, complexo, né, quando a gente faz um trabalho interinstitucional, principalmente envolvendo, né, instituição internacional que se compromete com fazer análise, mas aí a gente tem toda essa burocracia, né, de enviar a amostra, né. Então, foi bem delicado, assim, foi um processo longo, assim, de papéis, enfim, né. Tem uma aluna que perguntou o que que é, explica um pouco como foi exatamente o quê? Não sei se era o que eu estava falando. Ah, tá, eu falei. Lorena, I'm just talking a little bit more about the protocol when we conduct here the sub-study. Então, na coorte, né, a gente tinha feito um segmento aqui aos 11 anos, né, que já estava planejado dentro da coorte de 2004. Todas as coortes, né, então, aos 11 anos faz um novo segmento. Então, o estudo dela foi realizado um pouquinho depois. Então, foi após ter feito o segmento de 11 anos, né? Veio o subestudo, então, estamos ali mais ou menos de 14 anos, né? E aí, como ela falou, teve toda uma estratégia, a gente não fez esse subestudo com toda, né? Como envolvia a coleta de amostra, né? Então, era um dos focos, era a questão da microbiota. Não se pensou em nenhum momento fazer a coleta com toda a amostra, né? Pensou em fazer uma subamostra. Só que aí se fez uma amostragem dentro da coorte, né, de tentar, não seria só uma amostragem geral da coorte, mas pegando um grupo que teria obesidade, né, para a gente ter grupos para se comparar, né, a gente forçou um pouco, né, ter um grupo com obesidade e outro sem obesidade para ter grupos comparáveis dentro dessa subamostra, principalmente porque seria um N só de 400, né. Então, o desenvolvimento em si, o protocolo, né, foi muito ok realizar, né? A gente tem uma clínica aqui dos estudos de coorte, né? Então, mas muito desse segmento, por ser um N maior e envolver toda essa parte de coleta de amostras, eles foram feitos em casa, né? Então, a gente tinha pesquisadores que iam nas casas, então foi em torno de 400 adolescentes visitados, né? Nessa visita, né, eles eram previamente contatados, nessa visita se aplicava o questionário, né? E aí deixava o potinho coletor ali para eles fazerem a coleta, né? E posteriormente o pesquisador ia lá novamente e coletava essa amostra, né, que eles haviam feito, realizado, né? Foi uma questão interessante nessa questão de propor essa coleta de fezes, né? De coleta, né? Porque como tu vai convidar o pessoal que a princípio está mais acostumado a ser investigado, a responder questionários, né? Quando começa a se propor a coleta de sangue, já tem que ter todo um cuidado, né? Porque nem todo mundo é confortável, ainda mais adolescente, com coleta de sangue. E quando a gente foi, então, nesse subestudo, propor uma coleta de fezes, se teve toda uma estratégia, né? Então, quando a Lorena veio para cá, que ela estava se organizando, já estava praticamente já realizando o andamento da coleta, né? Ela participou um pouco dessa parte da, da coleta. A gente fez várias estratégias. Então, a Lorena, por exemplo, ela fez um vídeo, né? Que a fazer toda a aplicação dos questionários, posteriormente, ela apresentava um vídeo, era um vídeo bem lúdico, né? Explicando por que que a gente queria coletar a amostra, né? Então, foi assim, a gente teve que fazer todas as estratégias, porque quando a gente está elaborando um estudo, a gente está achando super interessante. Ah, vamos agora, né, nessa fase, coletar a amostra, né, de fezes, mas a gente tem que explicar, né, para as pessoas por que que a gente quer analisar, né, as fezes, né, e o que que a gente vai, de fato, analisar, porque as pessoas podem, né, pensar que a gente vai fazer uma análise usual, né, de que geralmente a gente faz, né, quando a gente é criança, adolescente, né, para avaliar algumas questões, né, usuais. E a gente quer fazer uma análise mais profunda, né? E aquela análise que a gente não vai dar vai entregar o resultado em breve, né? Então teve toda essa estratégia assim de explicar, de fazer essa questão do vídeo, né? para ter a adesão, né, para que eles nos retornassem com a amostra coletada, né. Então foi bem interessante assim participar dessa etapa, né, de também ter participado desse subestudo, né. A minha participação maior foi a questão do QFA, né, que eu treinei ali o pessoal a aplicar o QFA, né. Deixa eu só ver o que a Lorena comentou aqui. Vamos ver a Ana aqui. Ana, professora, uma dúvida metodológica em relação à coorte para fazer essa relação. Foi necessário analisar a microbiota mais de uma vez? Não, então foi só uma coleta, tá? Foi só uma coleta que a gente fez. Então a gente fez a coleta da amostra, né? E claro, a gente aplicou um QFA, o QFA é retrospectivo, né? Então era um método assim que a gente sabia, né, dessas questões, né? De certo ponto, algumas limitações, né? Mas foi o que a gente fez, né? Deu para fazer também, né? Não dava para propor mais de uma coleta, né? Porque foi um subestudo bem pontual, assim, né? Não, a princípio não está se prevendo coletar novos momentos, né? De amostra, né? De fezes para fazer essa análise, né? Então foi só
Yeah,
esse,
I think
claro.
ideally,
Hum?
yeah,
Pode ser.
ideally, as with diet, like you want to do multiple 24-hour recalls, might similar want to do multiple fecal collection samples. It's not always feasible, right? It depends on the study, the budget, and all these the timeline that you have. But overall, when you do a QFA, like a FFQ, you base it on the premise that diet is relatively stable for these individual, and similar with the microbiome. So if we were comparing one same individual with themselves throughout time, yes, you have to like assess multiple time points. But because here we're just comparing one group to the other, and we basically know that okay, microbiome is relatively stable at after age 3 to 5, depending what you want to think. So you want to get the overall picture of okay, what are the characteristics of this individual compared to these other individuals. So that's why we only collected one. But yeah, research now is moving to like improved methods to assess the microbiome as well. And yeah, Juliana's participation in this project was very, very important as well.
Bom, que mais, pessoal? Mais outras curiosidades?
There was also the question of the protocol for collecting the stool samples. So the gold standard for collecting stool samples is yes, you collect it, you instruct the participant to immediately freeze it, and then you transport it in a freezer like state until you have to assess it. So this was one of the challenges when we implemented the fecal collection in the Pilates cohort, because we were aware that not everyone might have a freezer. Like the transportation will like increase cost like so much that might be able to like just assess like a few individuals where we are already assessing like a very few. So we looked into other options, and we found these stool collection method that allowed to store the samples at room temperature for over 2 years. So they kind of have this buffer that stabilizes the fecal sample. Of course, it comes with challenges, like there are things that you cannot do, like depending on your analysis, your type of the way that you want to analyze that data later on will affect like how you collected it. So if you want to do like specific qPCR or like targeting specific microbes, you don't have like the amount of stool that you collected because it's all kind of diluted in this buffer. But this was the option that we came up with for us, and that allowed us to had such a large sample size within like the microbiome research. It was considered large by the time that we were doing the study.
Tem outra pergunta aqui, Lorena, if you had any recommendation for diet before in the day before the collection or physical exercise for the collection of the tools.
Yeah. No, we didn't apply any. So we had a questionnaire that they were supposed, like they filled on the day of collection, saying like whether they took antibiotics, laxatives, any probiotics, any other medication on the day of collection or the previous week. There were also questions about the storage of the sample, like, oh, did they leave it in the sun? Did they leave it in the fridge? Did they put it like somewhere else, just to like kind of quality control? And overall, we didn't find any samples that we had to exclude because of those characteristics. But overall, they did not receive any, other than how to collect the samples, they did not receive any other indication on what to do before the collection. Like, don't change your diet, don't do this, or don't do exercise. No. Exercise also was a question part of like the lifestyle questionnaire, but that was like over the last year, so not really on the day of. And overall, this is all based on research. Like, we know that microbiome doesn't change from one day to another, just by that. So the main thing that will change it is antibiotic laxative medications, like right away, that will affect your microbiome. But if you do exercise one day, it doesn't really change your microbiome right away. It's like a relatively stable community. So you need to implement like a big change, like an antibiotic, kind of like to modify it drastically.
Bem, pessoal, mais alguma curiosidade? Acho que, então, no momento, acho que foram essas perguntas, né? O pessoal acho que não está fazendo mais perguntas. Deixa eu ver se agora tem mais um comentário, não sei. Não, foi só um agradecimento. Então, tá. Então, pessoal, então, novamente, assim, Lorena, muito obrigada pela tua participação aqui, pela tua apresentação, né? Foi bem interessante, então, agora, pessoal, então, indo agora para essas últimas aulas, né, mostrando essa interação, essa análise mais complexa, né, de quanto que a gente pode explorar o microbioma também em estudos epidemiológicos, né? E até vendo, né, que é bem complexo, não é uma coisa tão simples e às vezes não vai bem exatamente conforme, né, as nossas hipóteses, né? Mas enfim, espero que vocês tenham gostado. Como a Lorena falou, esse estudo dela já está no pré-print. Se vocês quiserem, a gente pode encaminhar, eu posso encaminhar ali para o pessoal do fórum, para vocês terem mais detalhes, né? E agradecemos, então, Lorena, mais uma vez, tá? Pelo tempo, pela disponibilidade, ela que também está num fuso horário, ele não é muito grande, mas é um fuso horário, né? Que a gente tem que acabar se organizando para poder participar aqui da apresentação, tá bom? Simone, não sei se também tu queria comentar alguma coisa, fazer uns últimos agradecimentos.
Tudo ok, Ju, tudo ok. Thank you,
Então tá.
Lorena. Thank you.
Ok, então
Thanks
tá.
for the
Obrigada a
invitation.
todo mundo. Obrigada, então, a todos aqui, porque estiveram com a gente, né? Uma menina falou que quer receber. Eu encaminho também para o pessoal do fórum ali. Tá bom? Ok, so thank you, Lorena. Thank you, everyone that was here. And see you next Wednesday.